Again, thank you all so much for joining us tonight. Tonight's event is brought to you by MetaVet and MicroGen Vet. They have a really great and unique new comprehensive diagnostic screening tool that I think you guys are going to be excited to learn about. And I just really, first and foremost, want to thank our speakers for joining us tonight. I think you've helped us gather an amazing turnout, and we're really excited to see what you all have to say. And I think the best thing for us, Dr. Reed, is to go ahead and get started and let you start on with your presentation. So thank you so much.
You bet. Yeah, we need to get through what I have to say because I'd like to thank all of you for for participating, for coming on and listening. It's a kind of exciting thing, and we've been really, Dr. Barr and I have been really enjoying the opportunity over the last few years to work alongside, the development of this use of this new technologies. And certainly my part of this introductory is going to talk a little bit about innovation in diagnosis of equine diseases. There goes innovation, obviously it's something, a word that we're all familiar with, the action or process of innovating. A lot of neat synonyms from simple words like change, alteration, revolution, but informally it's a shakeup. And I think that in our profession, there are a lot of shakeups that are ongoing, but development of new diagnostic methods, and particularly use of this next generation sequencing is one of the big ones. As we get started, this is a quote that I remember from and I remember hearing Dr. Jim Kaufman deliver it. And it really, I thought, is appropriate back then and now to remind all of us that as practitioners and researchers, we're here for the horse and the medical and surgical needs peculiar to that species. And, throughout our work, we have to maintain a high level of awareness about why we do research on equine diseases, so that we don't lose our way or our bearings in the midst of rapid change. And things are rapidly changing, both in the way we do things as well as in the complexity and understanding. And certainly the high throughput technologies that next generation sequencing represents is something that is very complex and a rapid change. And all someone has to do is just open any newspaper or listen to the news, and discussions about new technologies, everything from CRISPR, which is a genome research tool or engineering tool that people think is going to be able to divide up the whole genome of whether it be human, mouse, or horse, and help us solve complex diseases. From my personal point of view, with a keen interest in neurologic diseases, we know that there are many monogenetic diseases that we're sort. But here we're looking at this as a diagnostic technology to help us understand organisms, bacteria, fungi, viruses that might be, involved. But one of the most notable medical innovations in the past decade is this next generation sequencing. And this is a technology that makes it feasible to comprehensively consider the genetic component of any medical illness. But more important and more germane to this discussion is how it might help us solve many of the sort of diagnostic odysseys that are in front of us. When we see a horse that has clinical signs that are similar, but yet the etiologies may be quite diverse, well, sometimes that might be due to genetic disorder, and at other times it may be due to some acquired etiology. And helping us sort through that information is a really, really critical thing. And just a couple examples on the human side. Here was a 14-year-old boy who had developed encephalitis and had continuous swelling of the brain. And doctors had done all of the routine diagnostic methodologies that they could to sort through to cause, determine what was the underlying cause of his encephalitis. And they finally went to the parents, and even though there was a lot of anxiety about trying a new technology, they chose to use this next generation sequencing, and sure enough, they were able to identify the underlying cause and end up with a successful outcome. In another example on the human side, here was a situation where an individual had a replacement joint. And in so doing, everything had seemed to go along for a while. Well, then he developed an infection 3 years after the original surgery. And they did all of the usual things and had to do several revision surgeries to help correct the problem, but they couldn't figure out what the underlying bug was. Well, then as they searched further, they used the next generation sequencing, and they identified Streptococcus canis. And then the owner commented that he had had a dog scratch, that dog, his dog had then licked the scratch, and this bacteria is in the mouth of dogs. And so here was a pathogen that no one would have ever suspected that, they were able to sort out, treat, and then eventually they could go back and redo the original surgery. So it's the methodologies and the cost. Some people worry about the cost, and Bonnie and Bimbo will talk about that more specifically. But again, the upfront cost of doing this has to be weighed and compared to what happens if you're doing multiple culturing, and particularly if you're doing multiple culturing, aggressive treatment, and not getting anywhere. Perhaps you don't have the right organism. But this can be useful to identify unsuspected or previously unknown pathogens that might cause infections in man and in many body systems. And it can really help clinicians select a more targeted treatment or better understand the cause of a treatment failure. Because whilst you might not be exactly the same as doing culture and sensitivity, what you can see with this, and you'll hear more about it as we go through the evening, it'll identify resistance genes so that you'll be able to know, well, perhaps the bugs that are causing this are not going to be sensitive to the usual antimicrobial agents. And, finally, it's likely to help us develop new and novel ways to deal with diseases in horses. Traditional culturing diagnostics has been the cornerstone of veterinary medicine for decades. And the adoption of these DNA-based technologies in human markets, it's changing the paradigm, giving us a lot of shifts and directions. So even though right now culture and sensitivity is sort of the gold standard, there are a lot of challenges there. For one thing, and we're going to hear very soon from Dr. Welker about biofilms, up to 80% of all infections are biofilm related. Only 1% of known microbes grow under today's culturing diagnostic tools. So again, we've got the challenge of the methodologies that have been around for a long time might not grow the organisms we need. In 30 to 35% of cultures, there's no growth, when in reality, there may very well be something there. And then we've also got the big consideration about aggressive bacteria that are arising from overuse of broad spectrum antibiotics in all species. So an understanding of biofilms, their impedance to treatment as that grows, and the absolute need for rapid and accurate identification of the important organisms causing a problem, as that becomes more and more critical, not that it hasn't been there for a long time, the need for new technologies like DNA sequencing and high throughput methodologies is going to be critical. So there are many advantages of that next generation sequencing has over traditional microbial diagnostic tests. number one, it can do a lot of unbiased protocols, so you don't have to go in there with a preconceived idea or with set primers for PCR. It's going to be more broad spectrum. It has the ability to detect fastidious or non-culturable organisms, and it has the ability to detect co-infections in biofilms. As I started at the beginning, things in our profession are changing rapidly. New techniques are sometimes complex, and they often cause us to have a little bit of pushback. But I think MicroGen Vet is an important company for all of us to get behind, because now we're ready to change and fight the dogma of what is the gold standard and what are new ways that we can get to answers to provide solutions. The niche of this company is to help veterinarians understand and utilize next generation sequencing. And with that, Dr. Welker is going to start telling us a little bit about uh, traditional culturing and about biofilms.
Thanks, Steve. Let me get control here a second. Hang on. Well, thanks everybody for showing up today. I kind of reiterate everything Steve said about the importance of this technology. My part tonight is to try to explain the impact that biofilm might have on our day-to-day functioning. I know that what I've come up with as far as biofilm, because I'm a 1980 graduate, I don't remember them talking about biofilm. If I reach back in the literature, the first discussions about biofilm was in the 70s. So I didn't hear about it when I was in school. And I can be honest with you, it probably didn't even come to the front until in the last few years for me anyway. But when you read about it and look at it, it's a very exciting topic to think about when you're trying to treat diseases anyway. There's two things I want to try to cover today. One of them is the limitations of culturing, which was also an eye-opener for me as I read through the literature about the limitations themselves. And the second thing is the hiding out in biofilm. So we want to discuss what biofilm is and what it does to our ability to manage cases. The first thing is, it's been since 1882 when they came up with the auger plate that culturing began. And since then, by the way, not many changes have occurred in auger plates. I'll show you the reports that they came out of. But in order for us to grow something up on a plate, the nutrients have to be right, the temperature has to be right, the pH has to be right, the organism has to be alive and growing and in a planktonic phase, which means that it is single cells. It's been the standard for 140 years. I mean, I get out of school thinking if I want to find out what's causing a disease process, I take a culture, I submit the culture, I get my results back, and I go to work expecting some results. The problem is we don't get cultures back all the time. It's to a point in my career where turning in cultures is almost anticlimactic because I don't anticipate a high percentage of the time getting anything back. And then when I do get it back, the sensitivity patterns don't match up to what I'm having to do in the field anyway. So here's an article from 2015. That's pretty current. And they were talking about the fact that there needs to be a revolution in media. This is 2015, remember, and they discussed the same thing about the fact that in order to grow organisms up, there had to be suitable media, and that media had to take into consideration things like nutrients, pH, osmotic conditions, temperature. And because of that, nearly 99% of the bacteria cannot be cultured by today's standards. The good news is, what they reported was that they're expecting some resurgence in the interest in growth of media, so that all of a sudden we're going to get all these extra medias that are going to come out to help us to grow them up. So for all the concern about that limitation is that they have to be alive and growing. You can't pick them up if they're already, if you pick up an area that the organisms are not alive at that particular moment. You have to have the right media to grow them up. There can be no previous antibiotics, as we were all taught. You got to take them off antibiotics for a period of time in order to try to get a culture back. And they have to be planktonic, meaning, again, they have to be single cells. And when we go into this next topic, we'll see what we mean by single cell versus being in clumps or in a biofilm situation. So we have to have all these things right in order to grow it up. and all of that for a return of 1%. So it really is, for me personally, as a practitioner, it helps me understand why maybe I've got a kind of a bad taste in my mouth about spending somebody's money on a culture, because a lot of times you get information back that's not real, or information that doesn't help you out anyway. So they're hard to culture, and even if you can culture them, over 80% of those infections are in biofilms, as Dr. Reed said. And biofilm is really a fascinating topic when you start talking about infectious processes anyway. So what we're talking about is the fact that these individual cells or single cells are the only ones that we can culture. Once they move into this biofilm situation, they are not culturable by the technology that we have today. Not to mention the fact that this biofilm that we're going to talk about is also a protective mechanism for the organisms, and so that our treatments may not work. Or the biofilm is an organization or a colony of bacteria and fungus growing together as a unit, and them each contributing something to the biofilm that helps protect it against things in the environment, or things that we might throw at it, or things that the body might throw at it. So biofilm is a preferred way to live by organisms. I think that was probably one of the first things as I was reading about biofilm that's important. I always looked at infections as being bacteria running around by themselves, but in reality, because the bacteria know that they're going to survive longer in a multicellular community, they prefer to live this way, and they're going to try to go that route. It gives them better defenses against anything that we can throw at it, or the body can throw at it, or the environment can throw at it. All these things can be defended against if they work together as a unit. So think of these organisms as coming together as a community and living cohesively and being able to fight away anything that we throw at it anyway. So I want to talk about the life cycle of the biofilm. The first thing that happens is once an organism can find a surface that it can attach to and can attach firmly, it will multiply. And once it gets to a certain critical quorum, it begins to put out molecules that calls in anybody in the surrounding area that wants to come and join the community. So these biofilms then begin to grow and survive together. These molecules that they send out upregulate the biofilm and the organisms to produce genes that will produce this material that will become protective for the cells or the community itself. The biofilms themselves, that's the other thing that we got to deal with, is all biofilms are not the same, because it depends on which organisms are in the environment, what the environment is like. But what that does is that means that the biofilm will change its defense characteristics, and that's going to also impact our treatment approach if we can ever figure out a way to get through that biofilm. So mature biofilm is, by the way, 85% of this extracellular polymeric substance. So most of what you see, this snotty material, is predominantly this material that's going to protect the organisms. Only 15% of it is actual bacteria. These polysaccharide strands, or DNA, is variable depending on the organisms, like I said, and the nutrients and the environment that they're in. So again, it's going to be a real variable. And on the surface, we can see these as that slimy material that you see on a wound. That's the actual biofilm. This does not have to be external. It can be internal. It can be in different organs. So it's not something that we can always see. The next 3 slides just give you an example of what organisms might do in the biofilm. Here's an example of Staph aureus producing a polysaccharide that, in the absence of this polysaccharide, the neutrophils and the macrophages can clear biofilm easily. But if that polysaccharide is available, then it can't get to it to make that happen. Here's another one you pick out of the literature talking about how beta-lactamase accumulates faster in this substance than the beta-lactam antibiotics can diffuse. And so if it gets there first, it's going to break down those antibiotics and they become useless. Again, this goes back from me as a practitioner of, why do I get a sensitivity that says this antibiotic will work, and then I put it into the animal and it doesn't work? So it's just helping me to understand what might be going inside the body. The other thing is that most of the EPS is positively charged, and those will adhere to aminoglycosides and sequester them away. So again, I'm just trying to show you how different organisms might contribute to the overall defense mechanism of the biofilm itself. Think of it as a group of bacteria, not just one single thing. Anatomically, the biofilm is divided into outer edge, which is where you're going to spend most of your time looking at it, because that's where most of the activity is. There's the mid portion, which is metabolically active, but there's not a lot known about it. Then there's the base, which is metabolically inactive and not culturable. The one we want to look at is the outer edge, because that's where what they call seed dispersal mechanism takes place, meaning they go from this biofilm, they get dispersed as planktonic organisms. That's the only time we're going to be able to pick them up and grow them on a culture, by the way, but that's how they spread around. The peripheral, or the outer edge, is the most active part of it anyway. The fence mechanisms of this outer edge prevents toxic substances from penetrating it. Here's two examples. They had biofilm treated with hydrogen peroxide, had no penetration within 50 minutes, and a Pseudomonas biofilm treated with glutaraldehyde had no penetration within 200 minutes. So this is fascinating to see how these organisms are trying to protect themselves from anything that we can throw at it anyway. These detached fragments are the part that's seeding the environment. And the discussion is whether that's a programmed reproduction or that's a result of physical forces. Something is causing them to detach, and that's how they get spread around to different areas anyway. The thing that scares people most about biofilm is that it has the ability to reproduce its entire mass within 24 hours. So you can disturb it as much as you can, but if any of these fragments are left over, it'll just reform someplace else. So it's a constant ongoing activity. The mid-body, like I said, is primarily geared toward protective responses. Not as much is known about that as it is about the outer portion. And then the base is pretty much inactive, not culturable. They don't show any metabolic activity. But the detached fragment, like I said, is the culturable part. That's where they have to be in order for me and you to pick them up, put them on a plate, and grow them up. If they are not in that form, we can't get to it. So that's pretty important to know. The facts about biofilm that I want you to take away is that 80% of infections are biofilm related. Again, that's easy to see on the outside on skin wounds and stuff like that, but that can occur in other organs of the body. And so it's important for us to kind of think about what biofilm might be doing to interfere with our ability to take care of it. 85% of the biofilm is the EPS, which is the protective part of it. It provides the defense mechanisms for the colony. And remember, the components of that biofilm matrix is going to be dependent on organisms and the environment nutrients that they have available to them. So it's going to be all over the place. The discussion has to, at some point, talk about how do we get rid of biofilm. And because of the fact that there are so many different kinds of biofilm, it may be impossible to come up with a single way to get rid of it or to combat it anyway. The outer portion is the part that's seeding out. The other things I wanted you to know, it can form in a matter of hours or days. Some of the original stuff that we talked about suggested that it's only present in chronic infections. If you read through the literature, you find where they found biofilm forming in as little as 2 hours. So I don't think you need to look at it just as a chronic problem. It could be occurring pretty quickly. Remember that it can replicate itself within a 24-hour period of time. And the biofilm community protects it from all the things that we or the environment can throw at it anyway. So pretty heavy duty stuff. You can run, but you can't hide. That comes into next generation sequencing because it's not dependent upon those some of those factors that we talked about. So from then until now, like I said, culturing's been around for 140 years. You have a 30% chance of catching planktonic microbes. It's only 1% of all known microbes are culturable. That's kind of a small number. I mean, again, I've been doing this for a long time and I didn't realize how puny that culturing could possibly be. PCR came around, but PCR is dependent upon primers. Primers are expensive to make. They are very sensitive. And if we had a PCR for every organism out there, that would probably be the cat's meow because it's a lot quicker. But then comes along next generation sequencing. They've identified the DNA fingerprint of over 50,000 microbes now. I'm pretty sure that most veterinarians can only enumerate maybe a couple of dozen organisms. There's 50,000 organisms that they've already found. It would be very interesting to find out how those might be contributing to some of the disease processes that we have. More than 500,000 human diagnostic tests have been performed in the last 11 years. Dr. Reed alluded to some of those. The very interesting about next generation sequences is with one sample, you get aerobic, anaerobic, and fungal information, as opposed to having to specifically request different types of cultures for different things. All of this from a single request. That's pretty fascinating. So when we look at the cost of next generation sequencing, don't forget about all the information you get that we're not going to get from sending in any one of these single requests on its own anyway. It's not affected by temperature, time, or the prior use of antibiotics. It doesn't appear. So those are all very positive things about next generation sequencing. It does not require the organism to be planktonic. So we can find this in biofilm without having to disrupt the biofilm. It's 99.9% accurate. It identifies over 50,000 species. It gives you a full landscape, not just what's culturable by our technology, which is very fascinating. And it's going to throw some wrenches in your work when you're trying to deal with this, because you're going to come up with all kinds of new organisms that we're going to try to have to figure out what their contribution to the disease process that we're looking at. It's not affected by prior antibiotic usage. Biofilm is not a problem because we're looking at DNA. It doesn't matter whether it's alive, dead, or moving or not moving. You'll get your initial results in 24 hours, and then you'll get second level results in 3 to 5 days. And you're going to get antibiotic resistant gene reports. That's going to be a common question. Where do I get my sensitivity? Dr. Barr is going to talk about this stuff in more detail, but the point is, this is something you're going to have to get used to. This is a new paradigm. Like Dr. Reed said, you have to think outside the box. And when you do, you're going to find out how exciting this new technology is. The kits are available through distribution, by the way, so they're already out there to get started with if you have the mind to do that. With that, I turn it over to Dr. Barr. Thank you very much.
Do I have it?
Go ahead and request it, Dr. Barr.
Oh, do I still need to request? Okay, I thought, okay, sorry, all righty.
Okay.
All right, well, thank you everyone for joining us this evening. I'm trying to get on the camera here. Dr. Reed, all right.
Yeah.
Okay, good.
You don't need me.
All righty, so I'm gonna, hopefully this is gonna advance. There we go. So what I'm gonna do is, for this last section, I'm gonna just briefly touch on identification of pathogens. I think Dr. Welker and Dr. Reed talked quite a lot about this. Then I'm gonna go into the respiratory study that we performed here, in which we took, and this was on equine samples, which it was just comparing the results we got from next generation DNA sequencing to traditional bacterial cultures, go over a few case examples, and then just kind of look at the next generation sequencing MicroGen vet, the information that you can get when you send a sample, and kind of and just being able to pull out all the information that is available from the report. Oh. Anyway, sorry. So just as I think Dr. Welker talked about this quite a bit, you know, we all grew up and when we went to veterinary school, we all learned just about the traditional bacterial culture, which identifies bacteria based on morphology and biochemical properties. So you have an enrichment media and then a biochemical identification with an antibiotic sensitivity. I guess my arrows got a little messed up there. There are advantages and disadvantages to traditional bacterial culture. Advantages are it is inexpensive unless you are submitting multiple samples. Do get an antibiotic sensitivity. Results are available most often within 3 to 5 days. We're all comfortable with interpreting these results. Disadvantages is slow growing, clinically significant organisms may not be identified, which oftentimes this includes anaerobic bacteria. And then sometimes special media is needed to culture specific pathogens. And then prior antibiotic administration can affect these results. Molecular diagnostics has been used in research for years. And with this, you identify the organism based on genetic or proteomic properties. And the two most common methods are polymerase chain reaction, in which an individual organism is targeted, but you will need a specific primer to identify that organism. And then the other method is DNA sequencing, and this is methodologies to identify massive amounts of DNA from a sample. And you extract all that DNA from the sample and you match it to a library database. The DNA or the next generation DNA sequencing, it's basically a two-step process. You have amplification of the organism DNA using a PCR, and then sequencing all of those fragments. And the most common method that is used is a ribosomal nucleic acid, also an rRNA platform. And with this platform, they are looking for highly conserved regions of the gene. So with bacteria, it's the 16S, and with the fungal organisms, it's an internal transcribed spacer. Advantages of the next generation DNA sequencing: it surveys the full bacterial landscape, not just the culturable microbes. It identifies organisms that are slow growing or difficult to culture. Prior antibiotic administration does not affect the results. Samples are not easily affected by time and temperature. Biofilms do not affect the results, meaning if there is a biofilm there, the organisms are present in a biofilm. DNA, our next generation DNA sequencing is not affected by the biofilm, and at least identifies fungal and bacterial organisms. And I know on the human platform too, they're even detecting some viral organisms. Whoa, hold on. All right, sorry. Okay. And then this is just, Dr. Welker went over this in detail, and this was one of his slides, is once again, we need to think about move our focus of pathogens, and we need to understand biofilms. So going from that single planktonic bacteria that will cause an acute infection and will grow in a traditional culture, to thinking about that community of bacteria or the biofilm. Disadvantages of the next generation DNA sequencing: it can be expensive when you just compare it to a single culture. But on the flip side, if you're doing multiple cultures, that can add up also. And so, this cost, when you look at it that way, the cost does not really seem to be quite so bad. Biocides and lidocaine have a negative effect on the results. There's no antibiotic sensitivity, as we are used to looking at antibiotic sensitivities, but there is some antibiotic recommendations, and I think what I find very helpful are the antibiotic resistant genes are identified. And then also a disadvantage is just interpretation of the results. What is considered a normal bacteria profile? All these results significant based on the clinical signs. So I think this is where we just need to relearn, rethink the way that we look at these results. Once again, remembering though to look at the whole big picture of the patient. What is going on with the patient? Is there an infection there? On the human side, they have been using next generation DNA sequencing for diagnostics, for diagnosing problems for quite some time. The different types of disorders are chronic wounds, device-related infections, sinus infections, urinary tract infections, ear infections, orthopedic infections, and respiratory disorders. And just to highlight a couple of the reports in the literature utilizing next-gen sequencing on the human side. This is a report that was published about eight years ago. And it just basically compares the culture and the molecular identification of bacteria in chronic wounds. And they had 168 chronic wounds, and they took regular cultures of the samples and also submitted a sample for next-gen sequencing. And their results showed that they were 17 different bacteria were cultured, but when they looked at the samples, the results of the next-gen sequencing, there were 338 different bacteria were identified. So concluding that culture underreported the diversity of the wound microbiota and failed to detect the most abundant bacteria in the wound. And then this study looked at more of the economic side of it, so the cost of treating patients with a diabetic foot ulcer. And it compared 1 year in which the patients were treated with standard of care based on culture results, and then another group several years later in which they used biofilm-based wound management based on the results of molecular diagnostics, next-gen DNA sequencing. And they found that the wounds in that group healed faster, and then there was a reduction, about a 69% reduction in cost. So those patients got better quicker, which ultimately less money was the cost was lower to treat those patients. And then lastly, can't see the whole little box there, but basically this study reports post-op patients that had a joint replacement and ended up with an infection. And when they collected synovial fluid, deep tissue, or swabs, what they found were 60% were culture positive, where 89, and sorry, you can't see the 89 in the little box, 89 of those were positive with the molecular diagnostics or the next-gen sequencing. So I just wanted to talk a little bit on the study that we did here at Root and Riddle, and it was just kind of, once again, just kind of a study comparing what results we got with when we did a culture and next-gen sequencing in parallel on these tracheal washes from horses that had been evaluated with suspected lower airway tract disease. And these horses had various clinical signs: fever, cough, nasal discharge, respiratory distress, or poor performance. Tracheal aspirate was performed either percutaneously or through an endoscope using a guarded endoscopic catheter. And the sample was submitted for traditional bacterial culture, just aerobic culture only, and then the next generation DNA sequencing at MicroGen Vet. And the objective was just to compare these results. And this is doing this kind of opened my eyes to, wow, I mean, there's a lot we're probably missing as we're just doing traditional cultures. And then the other little part we looked at too is what impact did prior antibiotic use have on the bacteria identified. And the results, we had 82 horses that were sampled. The culture results, they were reported out with antibiotic sensitivity. The next-gen sequencing, we got the first level, so the rapid screening within 24 hours, and then we got the comprehensive or the level 2 within 3 to 5 days. And I'm going to go into detail about just kind of going over the reports and what is included in these reports in a little bit here. Next generation sequencing identified 136 different bacterial species, whereas the traditional bacterial culture only identified 25 different bacterial species. And those were just what your streps, I think Streptococcus zoepidemicus and E. coli were probably, the top two that were identified. A majority of bacteria identified with culture were also identified with molecular testing, and a majority of the bacteria identified by molecular testing were not identified with the culture. And then just looking at comparing the samples, the culture and the molecular testing, 59% of those samples were not in agreement with any bacteria identified. So I have the picture there is just of a report in which the culture grew as strep zoo and another bacteria, whereas on the level 2 from the next gen sequencing, we grew 4 different bacteria, and they were not the same bacteria that were identified. And then just briefly, 53% of the, or sorry, 53 horses. So more than half of the horses had a history of prior antibiotic treatment. And so those horses that were currently receiving antibiotics or antibiotics had been discontinued 24 hours prior to the procedure. The next generation sequencing identified 4 times more bacterial species than the traditional culture. So once again, this study was just to kind of compare how many bacteria would we get identify with next gen sequencing versus what we would see with the traditional culture. So next gen sequencing did identify more bacteria than our traditional culture due to all the reasons that have been discussed. Biofilm formation, slow growing bacteria not isolated, anaerobic bacteria, antibiotics, or selection of bacterial species in which antibiotics were not effective. Traditional culture identified common bacteria documented to be involved in lower respiratory tract disease, whereas the next generation sequencing identified those bacteria and many more. And then next gen sequencing identified bacteria in the face of prior antibiotic administration. We do struggle sometimes with which organisms are significant. And you'll see here shortly is the report from MicroGen. You get quantitative information, percentages, and to be able to inform you as which bacteria are a percentage of the bacteria that are there. And then the other question that sometimes is this too sensitive of a test? Are we picking up dead or non-viable DNA? And it is difficult to sequence degrading or DNA. So the answer to that would be as no. And also the quantitative information is important that is available on the report. So I'm just going to talk about just a couple case examples here. So this was a 2-year-old thoroughbred filly that had been treated for pneumonia for several days prior to evaluation. She had just been treated with Genocin. And the initial tracheal wash was performed. And at that point, we just did a culture. We did not do the next gen sequencing. And 2 common organisms were grown, or at least that we see through our lab, was a strep zoepidemicus and an E. coli. The filly continued after treatment, and I think this filly actually was treated with chloramphenicol because she was azotemic on presentation. So we just went the route with chloramphenicol. But still, she continued to have some low-grade fevers, so the tracheal wash was repeated. And at that point, the culture was a no growth, but we did pick up several bacteria on the it says pathogenesis report. This is early on. The company's name now is MicroGen, but anyways, we did pick up several different bacteria, and we were able to revisit her treatment, and she went on and did fine. And then this is another example where this was a kind of a chronic low-grade problem that this young quarter horse filly had, but she had been treated for a pleural pneumonia for several months prior, had continued to have low-grade fevers, and had several courses of several different antibiotics. Uh-oh. Ooh, sorry. The ultrasound, somehow that got left off here, but an ultrasound on evaluation, we did identify that there just seemed to be one focal area on the ultrasound that seemed to be what was, causing some problems. A tracheal wash was performed, and in this one, we actually did it through the scope and went down into the right bronchus to obtain our sample. Cytology did show us that there was a suppurative inflammation, but the trach wash itself, the bacterial culture, there was no anaerobes seen. We just an enterococcus was identified, and sample was submitted for next-gen sequencing. And as you can see on the level 2 results, that an anaerobic bacteria was identified at a pretty high percentage, and the filly was treated appropriately and went on and did fine. And then this is a fun case that it's a canine case. Arrow, who was a working, I think, police dog. He had been ill for quite some time. They spent a lot of money on cultures and PCR testing. And despite all of that, the dog still continued to deteriorate. They went ahead and submitted a blood sample for next-gen sequencing. They identified several different bacteria and went ahead and, with the appropriate treatment, the dog responded nicely and is back into doing his job. And interesting thing with this is that they spent over 9,000 on cultures and PCR testing, and then just with MicroGen next-gen sequencing for less than $500, they were able to identify how the dog needed to be treated for much less. So samples to consider submitting: any body fluids, thoracic fluid, abdominal fluid, joint, CSF, transtracheal wash fluid, bronchoalveolar lavage, guttural pouch fluid, and then tissue samples, swabs of a wound, fecal material, whole blood, aseptically collected in EDTA tubes, remembering that biocides and lidocaine have a negative effect on the results. And the samples also can be stored in the refrigerator for a few days before shipment. So oftentimes, if I have something that comes in over the weekend, I'll just make sure that it gets put in the refrigerator, and then it'll get shipped out on Monday. And the samples go to MicroGen, which is, as far as I know, the only commercial lab that's doing the next generation DNA sequencing, and also has been involved with next gen sequencing for quite some time. So they, they're the leader, in this type of diagnostic testing. Thank you, Dr. Reed. So typically any fluid, send 3 to 5 milliliters, but you can send more. And then a tissue sample, they want a smaller size, a pea size. Swabs, you can send swabs. Usually I send multiple swabs, just moisten them a little bit with some saline. And when I say multiple, if I have to send swabs, I usually will send at least 3 to 5 swabs. You want to rinse the area with saline, or just wipe it with gauze. You don't want any pus or any mucus. And then no lidocaine. and no biocides. If you rinse the area well with saline. And on the MicroGen Vet website, there is instructions on collecting and submitting each one of these specimens. So this is just my how I interpret these reports and the information that I gather from the reports. There is a lot of information that can be gathered, and I think it can be overwhelming at first. But the level 1, the results, you will have those within 24 hours, and they'll contain the bacterial load, the real-time PCR, antimicrobial resistant genes, and then an antimicrobial profile. Level 2, you get those results within about 3 to 5 business days, and that is where you get the next-gen sequencing results, and you also will get an antimicrobial profile. Hope this works. So just to kind of look at this a little bit more in depth, the level 1 results, antibiotic resistant genes, they look for 10 different genes. And let's see, the bacterial load, you will get, if it's a tissue or a fluid, you will get a number of the load, or you will get, if it's a swab, you'll get low, medium, or high, and then you'll get your rapid real-time PCR results, which those are, because PCR, you have to have a different probe for each one. What they do is they have there's a set of 8 microbes that are traditionally found in wounds or as causing an infection, and this is the organisms that the real-time PCR will look for. So 7 bacteria and 1 fungal organism. So this is part of the report that you'll see when you get your level 1. You'll see on the top the resistant genes detected will be listed there. If there were no genes detected, it will say no genes detected. And then you'll see can everybody, hopefully they can see my pointer here. You'll see your area where there's the rapid screening PCR results. First, you'll see your bacterial load, low, medium, or high, and then you'll see if one of those predetermined organisms were identified in the sample. And then the level 2 results, this is where you'll get the next-gen DNA sequencing results. So the bacterial organisms will be listed in the order of the most prominent organism, and as a percentage, remembering that the 2% is the cutoff. So any organisms that were there less than 2% are not going to be listed, and then just the fungal organisms will just be on the list. And I'll talk about the antimicrobial profile here. Well, the antimicrobial profile is based on, this is just a profile. These are recommendations based on, and more of a reference. It's based on clinical studies in which that particular antimicrobial or class of antimicrobial has been shown to be effective against that species. So, I went the wrong way, sorry. So here's the level 2 report. Once again, you will have, still at the top, you'll still see your resistant genes detected. And then you will look down and you will see the organisms that were detected based on the next-gen sequencing. And you'll see next-gen sequencing here, and then here are your percentages for those different organisms. There's also a column in which they will identify if there's a gram-positive or a gram-negative organism, and then what type of organism. So whether it was an anaerobic, aerobic, facultative, anaerobe. And then the last section here is your antimicrobial recommendations. And once again, like I said, this is based on stuff from the literature in which that particular type of antibiotic has been effective against that or for that bacteria. And this is just oh, actually this will also be on the bottom, and this just is kind of a little key as antibiotic classes, and then the most commonly used examples. Now you do have to remember most of this is based on the human side, so there will be some antibiotics listed here that we may not be using on our veterinary patients. And then just to kind of show you, so this is what the report looks like. The previous slides were just parts of this report, but this is the report that you'll get. You'll get with the patient's name on the top, what the specimen, this particular one was a sinus, who the clinician was, and then your level 1 report, your resistant genes on the top, and then your bacterial load, and then if there were any bacteria identified or any fungal organisms identified. And then the level 2 report, so your resistance genes on the top, and then whatever showed up on the next gen sequencing, the percentages, and then all the other information. Okay, just to go over 2 more examples. This is a 19-year-old thoroughbred broodmare that had chronic unilateral nasal discharge. Dr. Tanner, our dentist here, looked at this horse and diagnosed a tooth root abscess with a secondary sinusitis, and there was the fluid sample was cultured, and you can see there's no growth. Fluid sample from the sinus, and then but the MicroGen, the level 1 report, there was nothing was identified on that rapid screening, and there were no resistant genes identified. But then when the level 2 came back, you'll see that several different anaerobic bacteria were identified. This horse was placed on, the appropriate, appropriate treatment, in addition to lavaging the sinus. And then I think this is the last case I was going to just kind of present is a 3-year-old thoroughbred gelding, a racehorse that had been initially treated in July for pneumonia, pleural pneumonia, had chest tubes, and then ultimately did need a thoracotomy done. The horse seemed to be healing nicely, had been off of antibiotics for a while. Then a draining tract was noted from the thoracotomy site. Antibiotics were started, no improvement or minimal improvement. So the horse was anesthetized, and that draining tract was kind of probed and debrided. And you can see a piece of tissue, probably some lung that was removed. And the culture was just a strep zoo and an E. coli. And based on this sensitivity, the horse had already been through a lot of these different antibiotics, but then the MicroGen, came up with several other different bacteria that we were able to just, treat the horse with systemic antibiotics for a brief period, and then more just of a local treatment. And the horse went on and did well. So just a couple of takeaway points, and then we can get into the question and answer period. Next-gen sequencing provides a complete picture of microorganisms at the site. You have to remember, though, you need to treat it like any other diagnostic test. You need to, in my mind, this is just another piece of the puzzle. And it's a great piece of the puzzle to have, but you have to look at the big picture. You need to, look and see, are there other signs, abnormalities that note that there is an infection present. And then based on all the information, go ahead and figure out the best way to manage the case. MicroGen Vet provides Their report provides a lot more information than just the microorganisms. And then just as I think is a good reference, as you're going through and you're looking at your reports, or you're just wondering what samples to submit, I think both the web pages or the websites for MicroGen DX, which is the human company, and then MicroGen Vet, both of them have some very good resources to look through. That is it. And I think we are right, Cody. We're going to open it up for questions for all three of us.
Yep, that's right. If you all have any questions, first of all, thank you, Dr. Reed, Dr. Welker, and Dr. Barr for your presentations. If you all have any questions, please feel free to use the chat feature, or if you're confident in your environment and don't have a lot of background noise, we welcome you to take yourself off of mute and ask a question as well.
So Cody, what's the cost of the test? The kit?
Yeah, the cost of the kit through distribution right now, it is available through some select distribution partners, is $209. Let's see here. I do have a question for Dr. Reed. I see, Dr. Barr, you're on mute, just to warn you. Dr. Reed, would this test work in possible identification of neurologic disorders like equine herpes virus 1?
Yeah, I think it would absolutely work because, you know, typically we look at spinal fluid, and most of the time we're doing PCR primarily or ELISAs on that, but this would be another source of things we could do. And particularly if you had a suspect bacterial meningitis, or we recently had, you know, a case that was, we had a lot of neutrophils in there and we could not sort it out. And, you know, again, it ended up being Lyme at postmortem, unfortunately, but I think this would be a very useful test, particularly in a fluid like that where it's not typically going to have, you know, a large amount of material, bacteria, virus, whatever, in that spinal fluid. Certainly when you're looking for herpes virus PCR swabs, again, you know, you're going to get a good probability on that. But again, MicroGen would get it on their level 1 testing.
But it's not available yet. The viruses, at least on the veterinary side, as far as I know. But yeah, they're doing a lot of viral testing on the human side.
So kind of along the same lines then, what do you feel about this as a potential diagnostic for EPM?
Well, we haven't done, you know, anything. And I think as far as looking at protozoans, right now the focus has been on bacteria and fungi, but any, you know, microorganism, you know, they could detect with this. So as a newer diagnostic test for protozoal myelitis, we haven't yet done anything. So rather than jump up and say, oh, absolutely, although that's my belief is that it would likely work very well, we haven't done that yet. So that's a fair question.
Moving on to the next one, and this one might be a question for the MicroGen vet guys, if you all are available still. And I think it is, yeah, does NGS also report plasmid DNA? Basically, if so, can the results be coupled with bacterial species of origin? Wondering if there's a plasmid DNA bank yet for, you know, encoded toxins, antibiotic resistance, et cetera. Anybody who might know about that one, please chime in.
Hey, Cody, it's Kurt here. We can definitely look into that. You know, I can definitely talk to the PhDs in the lab, but I do not know that. But we can get back to them on that for sure. Yep.
Yeah, that's from Alessandro.
This is a good question here. I assume a clinically normal, especially respiratory horse, will show many organisms. How do you interpret the results? And is SAA, serum amyloid A concurrent tool in most cases for you?
I definitely I would SAA is something that I do on all my cases. And once again, I think it's important to I think it's important to look at the big picture, make sure you are treating a horse that you are suspicious, or you're testing a horse that you are suspicious that has an infection. Yes, there are normal wait, sorry, yeah, microbiota. I'm trying to read the question and answer it, which is not a good thing. There's normal, you're going to pick up normal bacteria. I mean, especially the trachea is not going to be sterile. So there's going to be normal microbiota. And that's why I think it's important to look at the big picture and know, the horse has an infection. We're worried about, that this is an infection, oftentimes something chronic that we're trying to treat. And I think that can be challenging, as what is normal and what is not normal. And I think that's where you have to also, once again, look at all look at all your other diagnostics. And SAA, if I had a horse that had an abnormal high SAA, and had several organisms on the trach wash, I would say yes, I would go ahead and treat that horse, because I'm sure I'm looking at that horse because it has some respiratory signs.
And I think you got to remember that when you get those reports back, they are going to give you percentages or numbers of what organisms are there. So even if they are opportunists or bugs that you expect to be there, but they're there in excessively high numbers, you're going to be strongly suspicious that that's may very well be a part of your problem.
You're probably going to do that. You're probably going to do that in conjunction with cytology anyway. You're going to see neutrophils.
Right. Yes. Good. Yes. Good. Very good point there. Yes, Dr. Welker.
And Cody, just a broad point from MicroGen Vet. This is Ramey talking about, in the big picture, this is a relatively new application in the veterinary medicine world. So what we're looking for is a broader adoption so that we can gather more data, because that's really what you need to identify what a normal environment is going to be based on species in the system. We're going to have to gather that data over time. That's just what's going to happen.
Yep, and I think let's see here. I do have an answer in regards to the plasmid question. Dr. Chong, thank you for the assistance here with that. And he brings up the point that, yeah, as a NGS is only going to look at 16S ribosomal RNA or ITS or whatever primers are used, and it would not detect plasmids in its current form. So just kind of summarizing that for everybody. Dr. Welker, got one here for you in regards to biofilms and dominant pathogens. If left undiagnosed or untreated, do biofilms ever take over dominant pathogen, changing or mutating the outcome? I'm trying to, if left undiagnosed, read that again here, so on. If left undiagnosed and or untreated, do biofilms ever take over a dominant pathogen, changing the biofilm? Essentially, I think is where the question is going.
Yeah, so like there's multiple organisms and the primary organism is overwhelmed by the secondary organisms.
That's right.
I don't know that I've seen any evidence to support that at all. So I don't think so, but I'd have to look that up too. I don't think I've seen anything in any of the references that I've looked at.
And another question here for Dr. Barr, and this is in regards to no detection on level 1 and no detection in level 2. How often have you seen a report come back negative on an NGS report? Is that something that you have experienced, and what would you attribute that to? I mean, is it the fact that there is no longer an infection, or would you suspect an erroneous test?
Yes, I have had those types of reports. I guess it just depends on the case that I'm looking at. I guess, did I submit a large enough sample? I think that could maybe possibly be, be a reason. I guess, what is there not an infection there? I mean, I guess it just depends on what my other, what my cytology showed me, what my lab work showed me. Is it an error in reporting? I think that's where sometimes, you look at your bacterial load. If it's a low bacterial load, you may not be picked up. I guess the other thing would be too is if it's a very low, there it could be less than the 2%. Dr. Reed, do you have any comments?
No, yeah, I just think if you get nothing on the level 1 or the level 2, that, maybe there's either, as Dr. Barr already said, you got too small a sample, and that sometimes happens to us, or more likely it's not an infection. It's some other cause of inflammation that's not infectious.
Dr. Welker, in your experience, what do you see?
Well, I find it uncommon to have no growth in the samples that I've taken, but when I do the same thing. I assume that it's probably not infectious and look for something else. Or sampling. Sample error, I think, is going to be something to deal with until people figure out what they're going to need for different types of fluids or different types of samples. And like you said in your when you started off, you have to take the correct sample. You have to clean off the outside because they're going to pick up anything that's in the area, and you're trying to get down to the nitty gritty of what's real. So if you take a sloppy tracheal wash, you're going to get more than you asked for.
Right. And I think another, I've always thought that this would be great for, I do a lot of foal work. It's about all that I will do. So those foals that are septic, those newborn neonates that come in that are septic, and you don't, 33% of the time we'll get a positive blood culture, and you just know they're septic, but then on your culture you get, no growth on your blood culture. And this is, I would love, I think this is a test that it would be great to be able to do. And I have looked on the blood cultures, and those are the ones that have come up negative. And I think that's just, maybe there's a little bit too much background DNA. I'm not sure about that.
I think that the exciting thing about this new technology is that the more people that get into it, the more we're going to learn about it. And we're going to find out some of the answers to some of our questions that we have in ourselves or from these other people that are coming in. It's going to be fun to see the growth of it.
And even on the human side too, what they've been doing with this test.
Another question from the chat feed here. What is y'all's experience with NGS and MicroGen Vet in dealing with your joint infections? Kind of in line.
None of us do orthopedics, but certainly we have, I know several of the orthopods here have been quite interested, and were intrigued when this company first came to us to talk about it, because just like with things like in spinal fluid and other areas, oftentimes joint fluid or, as Bonnie just mentioned, blood cultures, it's rare to find a bug. And so this, I think in the few cases we've used, it's been quite helpful. But I think, again, we have limited experience, right, Bonnie? I mean, it's only been used in a few cases.
Yeah, I think the more we do it and the more we learn, how much fluid needs to be collected and submitted. I think it could be a great tool for joint infections.
I think the joint cultures are notoriously bad about growing things up, but also you couple that with the shortcomings of culture technology and then compare it to what we might get when we start doing this more often. That's the exciting thing about it.
Yeah, I would say, just from our perspective, we have been seeing a trend in more joint fluid samples. It is recommended, by the company, if you can, if ever possible, send as much joint fluid in as you can. They can accept up to 3 10 ml vials of joint fluid, so they can really handle a large volume of that. But it is also always recommended, if possible, and maybe I know this is probably a surgical application to get it, but if we can get a tissue sample from that source of maybe where that biofilm is, it's going to give us a better opportunity to identify what's going on in there. So it is all about the sampling with that. If you can't get a tissue biopsy of the synovium, they do also recommend on the human side, to swab the joint synovium and see what you can get. Let's see here. This is a good question. Is it likely that this advanced knowledge will lead to better bacterial vaccines?
I don't know if I know the answer to that. Certainly we see a few things where you've got a chronic, infection, and like particularly I think of where people have made salmonella vaccines, where they've had one particular problem on a particular farm and tried to do it. But I don't know the answer to that question, whether it's going to make a difference or not. But I suspect if you identified a unique organism that was problematic on one farm or one group of animals, then I would think it would be quite helpful. But
It's a very good question, a very interesting question.
I don't know.
And I
Yeah,
don't know the answer either.
From MicroGen Vet's viewpoint on that, we've had some discussions, and there's so many microbes that we didn't know about before that are present in these infections. So as we learn more about what actually is underlying some of these infections, I think that'll open the opportunity for vaccines to change to address those organisms, which we didn't know were there before. It's early days yet in that line of thinking.
And this is, again, maybe a question for Ray, and it might be a hard one to answer right now, but there has been a lot of talk about viral diagnosis. So this question is, how far away is this technology for equine for viral diagnosis?
Yeah, so I think Dr. Barr referenced that on the human side, they were doing quite a bit in the world of viral. We've not actually explored that with MicroGen DX, the laboratory, as far as translating some of that work over to the veterinary, to be honest. So we're probably a couple of years out before we get to that point. We're trying to tackle bacteria and fungal as the two starting points for the technology.
And that is it for our questions on the chat as it stands right now. If anybody else has any questions and would like to take themselves off of mute, feel free. We have a few more minutes here left before we have to, couple more minutes here before we have to end our call.
Cody, what about the sinus question? What method of specimen collection would you recommend for sinusitis?
Let me see. I think you even kind of touched that on yours too, about staying away from pus and when you're doing the sinus collection. I'm gonna send that one back up to Ray. Are you familiar with the sinus protocol?
Yeah, so I would recommend Dr. Brown, Virginia Tech. He's done dentistry, sinus, maybe 100 cases. He'll be publishing a paper on that later this year, I believe. And I'd probably have to get back to Dr. Brown to get the answer for you, to be honest. I'd just be fishing otherwise. That's the best way to do that.
Well, go ahead, Dr. Reed.
No, you go ahead.
I was just going to say, one of my cases was a horse with sinusitis. It was my own horse. And I'm pretty sure Dr. Tanner, he cultured some of the fluid from the lavage. I think Dr. Reed made a good point to me here. I think if you also could get a little piece of tissue, that also probably would be
Yeah.
beneficial, or maybe even swab an area or something.
Yeah, and one of the, I don't want to steal his thunder about it either, but a lot of his study is around the organisms he's finding in the sinus actually maybe originating in the mouth, right? There are organisms that are more commonly found in the mouth, but somehow they migrate up through the root canal system into the sinus and cause these infections. So there's a lot of connectivity he's saying, and I think that's what he's going to write about. So ultimately, he'd love to be able to see if you can do a swab or some simple mechanism to identify some of these earlier on before you have to drill a hole to actually get the pus out and actually identify it through tissue. But again, Dr. Brown has some insights on this. I think we can share once we talk to him.
But if you had a dental problem and a patent infundibulum, that would be the easiest way for something to get up through a tooth into the sinus from the mouth, you know, and that certainly in young horses would be something we worry about, patent infundibulum and that type of thing. Did you have is there another question, Bonnie?
No, that's it.
Right. You.
Hold on, I have you muted.
I just said it's been good to be with you and thank you.
Yeah, thank you all again. And again, special thanks to our presenters tonight. We really appreciate your time in joining us. And again, thank you all for turning out tonight. Really appreciate that. If MetaVet, our company, or MicroGen Vet can be of any service to you, obviously we got in touch with you via email. Please feel free to reach out to us. We will be following up this call with a with a little survey just to make sure that y'all are happy with the outcomes and presentations. And if there's anything else that we can do for you, please feel free to reach out. Thank you all.
Thank you very much.